is Rebecca. Natalie, Jack, Wilson, and I did our presentation on the paper about long non-coding RNA CCAT2 promotes breast tumor growth by regulating the WNT signaling pathway.
Breast cancer is described in the article as the most commonly diagnosed cancer in women, while metastasis disease is the leading cancer-related death. The article also explains how cancer progression involves changes in gene expression, including in long non-coding RNAs, which have increasingly been shown to regulate cell growth, apoptosis, cancer progression, and metastasis. Alterations in the long non-coding RNA expression structures and bindings interaction are frequently observed in human cancers and are often linked to metastasis, invasions, and patient survivals. The function of CCAT2 is a long non-coding RNA that does not encode protein. It regulates RNA whose abnormal expressions are linked to cancer development. The article states that CCAT2 was first discovered in colorectal cancer, which it was found to be highly overexpressed and promoted tumor growth, metastasis, and chromosomal instability. The role of CCAT2 in WNT beta-catenin signal pathway, its major regulatory pathway controls cell proliferation, differentiation, survival, and tumor formation. Beta-catenin is the central factor in the pathway. When activated, it enters the nucleus and stimulates transcription of the pro-growth gene as a CMYC and CCND1. It's enhanced by CCAT2 by increasing downstream oncogenic targets which promote breast tumor growth. CCAT2 expression is significantly upregulated in breast cancer tumors and cell lines compared to normal tissue cells. CCAT2 is linked to several cancers. It also contributes to breast cancer. It is widely unknown. It was unknown if CCAT2 is upregulated in breast cancer, if CCAT2 affects tumor cell behavior, and identifying the pathway of CCAT2 promotes breast cancer. The hypothesis is if CCAT2 promotes tumor growth and progression, potentially by influencing the WNT beta-catenin signal pathway.
Now I wanted to go into a bit of an overview or a roadmap of the study. So what the scientists wanted to do is they wanted to see if a long non-coding RNA, CCAT2, had any impact on breast cancer development, and mainly they wanted to see if alternating its gene expression could be used as a possible therapeutic in the future for patients with breast cancer. And so what they first did is they wanted to see the expression levels of CCAT2 within breast cancer tissues. So what they did is they used an RT-qPCR, which is a reverse transcript quantitative polymerase chain reaction, which this revealed it had high expression levels, which indicated that this could possibly be a reason for breast cancer to spread. And so what they wanted to do is to find a way to inhibit or reduce it. So they used an siCCAT2 and an siNS. These are small interfering RNA. NS is specifically non-specific. And so what they did is they transfected into the cells and to see if it had any impact. And it was revealed that siCCAT2 decreased cell proliferation and cell invasion. There was low expression levels and it barely spread. It did not migrate. It did not grow. And this was shown also through an MTT assay and a transwell assay. Now, another major thing is they wanted to see how it impacted the in the beta-catenin signaling pathway. And so they used the SICCAT2 and the SINS as well to see how it impacted the pathway. And it was revealed that the SICCAT2 had low activation, which meant it had low expression. This was shown between a Western blot and RT-qPCR and a top FOB flash lutefarase reporting system. Now that we got a bit of an overview, we're going to go ahead into a little bit more about some of the possible important experiments that were done. In the second most important hypothesis-driven experiment, Figure 5 tells us the WNT beta-catenin signaling pathway is impacted by gene expression. Figure 5b specifically is when the breast cancer cells were treated with SICCAT2 and or 10 micromolar of FH535. FH535 is a signaling inhibitor specific to this pathway. It was revealed that FH535 it only inhibits the beta-catenin part of the pathway, and SICCAT2 and the FH535 are both needed in order to impact the WNT part of the pathway. This is what causes the CCNDI and the CMYC expression levels to be reduced. This causes migration, invasion, and proliferation to decrease. This was revealed through an RT-qPCR. Now, the most important hypothesis-driven experiment in the study is Figure 4. It showed how gene expression impacted tumor growth. Specifically in 4C, the scientists put SINS or SICCAT2 into the new mice cells to see how tumor was impacted. This was then weighed after a couple of days to see how it impacted, where it revealed that cells with SICCAT2 reduced the tumor much more. Now we are going to go ahead and go into a little bit more detail about the second most important hypothesis-driven experiment.
So I'm going to go through the second most important experiment, which is shown in Figure 5b. This experiment examines whether CAT2 affects the expression of the WNT beta-catenin target genes, specifically CMYC and CCND1, which are both major drivers of cell proliferation. Our question was, does CAT2 regulate the expression of WNT target genes such as CMYC and CCND1? To test this, the researchers used a straightforward approach. They used normal control cells as well as they used CAT2 knockdown. They also used a WNT pathway inhibitor, which was FH535, as well as a combination of both treatments. The results ended up in knocking down CAT2 decreases mRNA levels of WNT target genes. FH535 was also seen to reduce expression, and the combination produced the largest reduction. For controls, our controls were the GAPDH control as internal reference gene. We also had the negative control, the scrambled short-strand RNA that served as the negative control, as well as the FH535, which acted as a positive control for the WNT inhibition. For the second most important experiment, hypothesis-driven experiment, for the conclusion, the researchers concluded that CAT2 is required to maintain normal expression of the WNT beta-catenin downstream genes. This means that CAT2 acts as a regulator of WNT signaling and pushes the pathway toward a more tumor state. For our category, for our classification, said it was a mechanistic expression experiment, as it directly looks at transcriptional output of the pathway rather than the upstream protein levels. To answer why this is a hypothesis-driven experiment, it directly tests a prediction made by the hypothesis, and it confirms that prediction and helps kind of bridge that gap between the CAT2 expression and the tumor promoting behaviors observed earlier in the study. And how it supports the overall hypothesis, again, it shows that CAT2 knockdown decreases the WNT target gene expression, and the experiment demonstrates that CAT2 operates upstream of beta-catenin dependent transcription.
As for our most important hypothesis, we determined that the most important hypothesis-driven experiment in the paper would be Figure 4, specifically Figures 4C and D, which investigate how suppression of the CCA2 long non-coding RNA can impact tumor growth and weight. The question that they investigated in this figure is, does the CCA2 long non-coding RNA have an impact on breast cancer tumor size? And if so, does treatment with a small interfering RNA change the impact of CCA2? The method they used was qRT-PCR, and then they collected and weighed tumor samples from patients. As for the results, Figure 4C shows that the tumor weight dropped significantly after treatment with the siRNA, along with a drop in relative expression of the CCA2 AT2 long non-coding RNA as seen in 4D. This experiment were first the nonsense siRNA, which serves as a negative control by providing basically what the results would look like if they were not treated with anything. Then, for the conclusion, the authors were able to conclude that the CCA2 long non-coding RNA is directly related to tumor growth because treatments with the nonsense siRNA showed significantly higher tumor weight and higher tumor expression than when treated with the interfering RNA for the CCA2 long non-coding RNA. We determined that this experiment was a combination of a functional experiment in the case of the tumor weight and an expression in the case of the qRT-PCR. Additionally, we were able to conclude that this was a hypothesis-driven experiment, as they were directly asking a question about what would happen if they inhibited expression of a certain gene and how that would impact tumor size.
So for our validation experiment, our goal was to retest the most important hypothesis-driven experiment in the study, which was the WNT luciferase reporter assay, but using a completely different method that still answers the same question in the paper. The original experiment, which was Figure 5C, showed that knocking down CAT2 reduces the WNT beta-catenin transcriptional activity using an artificial reporter system. While this was effective, it only measured the activity on a synthetic plasmid and not really on any real genes in the genome. So for our experiment, we did a CHIP qPCR assay, which allows us to look directly inside the cell at whether beta-catenin physically binds to natural WNT responsive promoters when CAT2 is knocked down. For our experimental design, we would perform chromatin immunoprecipitation using an antibody against beta-catenin. After immunoprecipitating beta-catenin bound DNA, we would run the qPCR for promoters of the well-known WNT target genes, which include the CMYC, CCND1, and additionally AXIN2. We would compare the promoter enrichment between the control cells, the CCAT2 knockdown cells, the FH535 treatment, and the combination of knockdown plus the inhibitor.
Now that we've established we'll be using chromatin immunoprecipitation in our experimental design, we'll go ahead and do the controls of the experiment. Now, these controls are very similar to what was used in the study. As the positive controls, the SI-CCAT2, this binds to the CCAT2 in breast cancer cells. This is guaranteed. We know it will impact expression levels. Now we'll also use the SI and S as a negative control, as it binds to some breast cancer cells and not all that will be the CCAT2 in the cells. This is a way to kind of compare between expression levels and determine what could be possibly a true value and influence. Now, of course, we'll also be using an internal control of a GAPDH. This is a housekeeping protein. It's the ideal standards. It's a way to be able to detect any change that could be possibly happening in the cell. Now we'll go ahead and look into the.
Here you can see the results for the mock-CHIP qPCR experiment for our validation. Figure 1A shows what the gel would look like with a ladder, the treatment group of the small interfering RNA, the nonsense RNA as a negative control, and the GADPH as a positive control, as it is a housekeeping gene in both cell lines. Additionally, we can see the percent input, which measures the relative percent binding of both small interfering RNAs in both cell lines. So, as for our conclusion and what the results mean from our validation experiment, we can see that in the validation experiment, it shows that the small interfering RNA binds to the beta-catenin promoter in the DNA protein complex, which can then be visualized by precipitating out the DNA protein complex and visualizing it in the gel, and then quantified using qPCR and analyzing the percent input. This particular experiment shows that the small interfering RNA has a strong binding to the promoter, suggesting that the CCAT2 long non-coding RNA does have an impact in the beta-catenin and Wnt pathways, and therefore inhibiting it with the small interfering RNA would have some impact on these pathways, and therefore potentially some impact on breast cancer and tumor growth, development, and other properties of breast cancer.